Okay, sorry for the technical issues here. What I want to do in this talk is propose a useful alignment-free proteome distance that can be used for distance-based phylogeny. And I'm actually going to narrow down this goal. There are multiple artifacts, obviously, that can affect any phylogeny. What I'm really going to be dealing with here is addressing the issue of long branch attraction. So sort of keep that in mind as sort of the narrow goal. I think unless we can address that, we're not going to achieve useful distance. So why distances? Why not, maximum likelihood, et cetera, et cetera? Well, if we think of distances, there actually may be some advantages. And I've shown at the bottom an ultrametric tree, and this is a species tree. Of course, pairs of taxa within this tree have gene trees with varying coalescence times. And to capture that, I'm not sure I'm getting the pointer, but why don't I just kind of do it narratively? I've made the coalescent time sort of fuzzy below. So what you can think of for at least some certain well-behaved distances would be that we're simply adding something to the sum of the two post-speciation branch lengths. Now, the cool thing about that is we make a tree from those distances. It should be the species tree, as long as we've sampled enough of the genome or proteome that we've captured the mixture of coalescent histories. Now, there are some problems. The sequence length requirements in principle might be prohibitive, possibly even bigger than, well, the genome. We might be able to bring those down by using covariance information. I'm really not going to go there. This has been sort of put into implementations like BioNJ. I don't think you can use BioNJ, and for reasons I won't go into, feel free to grab me and ask my reasoning here. But I think there are ways to go and deal with this potential sequence length requirement. Okay, now to the meat of the matter, alignment-free distances. How can we do it? Well, possibly the most principled way would be to sum over a set of alignments. And indeed, you could even do this with multiple taxa. This is exactly what the Bayesian program BALIFY does. They're trying to integrate over a set of multiple sequence alignments. This does bring in requirements for knowing something about a model of indel evolution, but you could argue that that's a really good way. Another way would be just to break things down into substrings and compare those substrings. That certainly can work. However, what I'd like to discuss is just using data compression. So this idea has been proposed before, and it's actually about 2 decades old. And the idea here is something I've shown up here called the normalized compression distance. And this is a very general distance between data sets X and Y. And all we're doing is looking at compressed file sizes to see the amount of additional the additional information of sort of putting these two together. So you could calculate distances between, say, translations of literature or something like this. This is very general, but we're going to use it to compare genomic data. Now, let's see. There are some issues with this, and they're shown here. So if we consider a true mammal tree, and this is, very schematic, primates and rodents should be united in a clade called Euarchontoglides. And this is quite well corroborated as a tree. However, rodents, especially muroid rodents, are a long branch. And I've indicated that with that dashed line sort of coming out of this cladogram to sort of focus you on high rate. When compression-based distance, when this simple NCD is used, notice that rodents shift deeper in the tree. They are probably being attracted to the long outgroup branches. Other problems with NCD: can't assess support using something like the bootstrap. Here's another thing: it's potentially very sensitive to compressor performance. You know, using gzip with the best setting versus using it with just, you know, a default setting, that's an undesirable behavior. And of course, it doesn't incorporate biological information. So how can we deal with that? So we can't use the bootstrap to assess support. So the idea I had here is to use a tiled format for the proteome file. So in other words, you create a file for both taxon X, Y, instead of simply concatenating them, just the whole proteomes. What you do is you take protein 1 from taxon X, follow it by its ortholog from protein Y, do then protein 2, et cetera, et cetera. This requires ortholog ID, but not multiple sequence alignment. For these tests I'm going to show you, I used one-to-one orthologs, but we may be able to extend this to more complex orthology relationships. This allows something analogous to bootstrapping, which is jackknife the orthologs. This does allow some means to assess support in a manner analogous to standard bootstrapping. It's jackknifing instead. Let's solve the problem of compressor performance. And this is an imperfect solution, but essentially what we're going to do is measure the NCD between taxon X and itself, and taxon Y and itself, and correct the NCD between taxon X and taxon Y using this information. This should render a value such that NCDC for corrected is much closer to 0 if the sequences of the proteomes are the same. Of course, it will be sensitive to indels. That's part of the strength, I would say, of alignment-free distances. Okay, this still does not incorporate biological information. It ignores amino acid properties, and it does not correct for multiple substitutions. The latter might be especially important because it will make the distances non-additive, which is probably the basis for the long branch attraction. How can we incorporate amino acid properties? Well, we can make recoded alphabets that group amino acids by, say, physicochemical properties. So for this Dayhoff alphabet, it's an alphabet of 6 symbols. And you can see the groups up there. So we would assign a 0 for all cysteines, AGPST gets a 1, etc. And we're combining this information because the amino acids are physicochemically similar. The one exception to this physicochemical property is this GC alphabet. I introduced that to look at state frequency changes. And essentially what we're doing here is we're grouping amino acids based on their codons. So FY, MI, and K have A and T bases for the first 2 positions. GAR and P G and C for the first two, and then in the middle it's one A or G or C and one A or T. So since genomic GC composition may have an impact on proteome amino acid composition, this could potentially capture that. Now, that's correct for multiple substitutions. It probably hasn't escaped your, or I don't know, it probably hasn't escaped your attention that NCDC for any of these alphabets is analogous to a p distance. So we simply use either a Poisson correction or more appropriately probably a gamma type correction. Now, how to estimate the alpha parameter in here, which captures among site rate variation in aligned distances, is unclear, and even its meaning, but a range of parameter values may perform well. We're just going to sort of leave the details of the estimation to the side for now. Let's test it. And the test I'm going to show you was done with mammalian proteins, 1,453 orthologs, same taxon samples as those previous studies that I boiled down to little cladograms. Okay, here's the concatenated ML tree. We are probably in a part of species tree space where, well, we are in a part of species tree space where the coalescent doesn't matter for the relevant clade, which is this rodent primate clade. Complete support, 100% bootstrap for all nodes except the one with a number, which you can see is a very short branch. But let's keep focused on the long branch attraction and the long branches to the rodents. If we use uncorrected distances, you see evidence for this non-additivity of the NCDC. Okay. And you can see that the rodents are placed as an outgroup. In general, there are longer, in relative terms, terminal branch lengths, and more importantly, more even root to tip branch lengths. This is actually undesirable because we're not correcting the branch lengths for the rodents enough to put them where they almost certainly belong. We correct with alpha equals 0.7, we get, well, a tree that looks much more like the ML tree. And as I said, that's been corroborated with multi-species coalescent methods and so forth. The red star does indicate one rearrangement within primates. This is a minor problem and it really doesn't go away. How did we choose alpha equals 0.7? Well, provocatively, the concatenated ML estimate of alpha is almost 0.7. But what I'd like to show you are graphs of different alpha values. And if we go at the left end of these graphs, we have very low values of alpha. At the right edges, we have high values of alpha, the Poisson correction, or no correction. Different colors are different alphabets. And what you can see is that as we move down in alpha, the support for rodent primate clade, Eugarcontoglires, goes up, and that's true for all alphabets. Unfortunately, support for marsupials goes down. You do see slightly different behavior for different alphabets, so we are capturing somewhat different information there. What's going on is that in terms of the marsupials, is that the relative branch lengths are quite different. You can see this very long branch to the wallaby. So when we apply lower values of alpha, we are sometimes breaking up marsupials. What exactly is going on there, I am not certain of. We are obviously capturing somewhat different information, and it could in principle be an annotation quality issue. So how do the different alphabets behave? They do, as I showed you, yield different support values. I just want to emphasize this by going back, and you can see, for example, the Dayhoff and Hanada alphabets being higher numbers in the Uarcontoglires, and actually the opposite relationship occurring for marsupials. Big point is we are capturing different information. Overall, that's the bigger point. We're capturing slightly different information from standard aligned distances or ML, which is, of course, an alignment-dependent methodology. Okay, I'm sorry, I got a little behind on this. I'm going to sort of wrap this up a little bit just to show you that we get a saturating function as we would expect by analogy with p distance, but you can see some outliers, again, emphasizing that we are capturing different information. And this is true, as you might expect, even when we transform the values. So the orange dots are transformed aligned distances. The blue dots are capturing transformed NCD. And of course, the accumulation is different for different alphabets. Anybody who wants to sort of go through details, feel free to grab me during the break so we can get back on time. And I'm going to skip over this. I'm sorry we got behind with the technical things, but just to summarize, I hope I can have convinced you that we can deal with long branch attraction, if nothing else. And I assume I do not have any time for questions. Okay.
So, questions for Ed?
Thank you.
How do you deal with the cases in which the ontology is missing in some species?
So how do I deal with missing orthologs? Great question. And the short answer is I only compare for each pairwise distance orthologs that are present in both. So this could also pull some additional information, whether it's a bug or a feature, into the distance. It's an open question, but that could be another source of additional information that's being pulled out here. But basically, I skip over things unless they're present in both. Great question.